Bismillahirrahmanirrahim. Assalamu alaikum dear students. This is Dr. Azam Ali from Department of Biochemistry, FRPMC. This video is a part of online practical series for the cardiovascular module. Today's topic is estimation of cholesterol. This video is divided into three parts. In the first part, we'll briefly discuss structure, function, and biomedical importance of cholesterol. In the second part, the principle, procedure, and calculation for the estimation of cholesterol will be explained. And in the third part, the estimation of cholesterol will be practically demonstrated to you. Cholesterol is the most important sterol in animals. Here you can see the structure of cholesterol. It comprises of a steroid nucleus having 4 fused rings. Attached to the carbon number 17 of the steroid nucleus is an 8-carbon side chain. Also, it has a hydroxyl group at carbon number 3, a double bond between carbon number 5 and 6, and methyl groups at carbon number 10 and 13. So this is how the cholesterol crystals appear under the microscope. You can see they have very sharp corners. And in this diagram, you can see the plaque formation in the arterial wall. It has been proposed that these cholesterol crystals cause rupture of this plaque, leading to thrombosis. Cholesterol performs several important functions in our body. It regulates the fluidity of the cell membrane. It can form acyl linkages with fatty acids. These lipoproteins are the transport vehicles for the lipids. Cholesterol is insoluble in water. Therefore, the transport of cholesterol in plasma is made possible with the help of these lipoproteins. Cholesterol is a precursor molecule for steroid hormones, vitamin D, bile acids, and bile salts. Now these are the structures of bile acids. Cholic acid and chenodeoxycholic acid are the primary bile acids, while deoxycholic acid and lithocholic acid are the secondary bile acids. These are the structures of steroid hormones. Among these, cortisol, which is a glucocorticoid, aldosterone, which is a mineralocorticoid, sex hormones such as testosterone, estradiol, and progesterone. Calcitriol, also known as vitamin D. Now you can appreciate the similarity between these steroid hormones and the structure of cholesterol, with the exception of this calcitriol, where the B ring of the steroid nucleus is open. Ecdysone is also a steroid hormone found in arthropods. Serum cholesterol levels. The desirable level for serum cholesterol is below 200 mg per deciliter. Serum levels between 200 and 239 are considered to be borderline high, while serum levels above 240 are considered as high serum cholesterol levels. Hyper and hypocholesterolemia. Hypercholesterolemia is the increased level of cholesterol in blood. Disease conditions in which cholesterol level in blood may be increased include nephrotic syndrome, obstructive jaundice, diabetes mellitus, hypoparathyroidism, familial hypercholesterolemia. In contrast, hypocholesterolemia is a decreased level of cholesterol in blood. The causes of hypocholesterolemia may be A-beta lipoproteinemias, hyperparathyroidism, pernicious anemia, and malabsorption syndrome. Now we proceed to our topic, that is estimation of cholesterol. Apparatus required for this practical include centrifuge machine, spectrophotometer, test tube holders, test tube rack, pipette and pipette fillers. The reagents required for this practical are ferric chloride in glacial acetic acid, concentrated sulfuric acid, cholesterol powder, and chloroform. Lieberman-Bouchard test. Now this is the test that we will do to estimate the level of cholesterol in a given sample. Its principle is cholesterol in the presence of glacial acetic acid and sulfuric acid gets dehydrated and form 3,5-cholestadiene. This 3,5-cholestadiene will further react with sulfuric acid to form colored sulfonic acid derivatives. And these colored compounds will then be measured with the help of a spectrophotometer to estimate the level of cholesterol. Spectrophotometer. It is an instrument that measures the amount of light absorbed by the sample. It consists of a light source, a lens which focuses the light on monochromator, and the monochromator will split the white light into different colored lights having a specific wavelength. Now this specific colored light will pass through the sample, and the light emitting from the sample will strike the photodetector. Different compounds transmit, absorb, and reflect light at a specific wavelength. This photodetector will show the transmittance, which is the ratio between light exiting from the sample and light entering the sample. If we place a colorless sample in the cuvette, then all the light entering the sample and all the light exiting the sample will be same. But if we place a colored sample in this cuvette, then the light entering the sample and exiting the sample will be different, which means some amount of light has been absorbed by the sample, which is colored, and some amount of it will be transmitted through the sample, and that transmitted light will be detected by this photodetector. Hence we can deduce that intensity of the color of the sample is the measure of concentration of the colored compound present in the sample, which means darker the color, higher the concentration of colored compound, lighter the color, lower the concentration of colored compound. Now the question arises that how a spectrophotometer will help us in estimation of cholesterol. If you recall, in the Lieberman-Bouchard test, we were converting cholesterol into several color derivatives of cholesterol. Thus, by measuring the concentration of those color compounds with the help of a spectrophotometer, we can measure the level of cholesterol in the given sample. Also, we will prepare a sample of cholesterol with a known concentration. Then we will compare the concentration of two samples, unknown and the known one. Method. Now we will begin with the preparation of sample. For this we will take 1 ml of whole blood and centrifuge it into the centrifuge machine for 20 minutes. After centrifugation, we will get the plasma, and the plasma will again be centrifuged in order to get a serum. So we'll take 0.1 ml of the serum and use it as a sample. Simultaneously, we'll prepare a standard solution of cholesterol. The purpose of this standard solution is that we know the concentration of this solution, which means we will take 100 mg of cholesterol and prepare a solution of 100 ml in chloroform. So in the end, we will compare the concentration of the unknown sample with the concentration of the standard. So students, after the preparation of sample and standard, we will perform the procedure. For this, we will take 3 test tubes and mark them as B, S and T. B stands for blank, S for standard and T for test. First of all, we will take the sample, and for this we will take 0.1 ml quantity and add that to the test tube which is marked as test. Then we will take the working standard, the standard for which we know the quantity and the concentration. So for this we will take 0.1 ml and add it to the test tube which is marked as a standard. Then we'll add ferric chloride 6 ml to all 3 test tubes and sulfuric acid 4 ml to all the 3 test tubes. After adding these reagents and samples, we'll leave these test tubes for 20 minutes and then measure the transmittance in a spectrophotometer for which the wavelength is set at 540 nanometer. Now, before measuring the transmittance of standard and test solutions that we have prepared, we have to first calibrate the spectrophotometer, and for this we'll first place the blank and set the transmittance to 100. The purpose of doing so is that we want to nullify the effect of these two reagents so that they don't interfere with our final reading. So now we'll move on for the demonstration of the entire process. Now I'll do the calculations, and you will use this formula: optical density of tests divided by optical density of a standard multiplied by 100. Now this table shows the relationship between transmittance and optical density, because the spectrophotometer has provided us the transmittance of standard and test solution. So we'll look for the optical density of corresponding transmittance that we have gained. For test, we will take 0.783, and for standard, the corresponding transmittance is 0.204. So we'll obtain this result, and for this sample, the cholesterol level is 383 milligram per deciliter. Thank you.